So today's speaker is Ammon Corl. Happy to introduce Ammon for those of you who don't know him already. So Ammon is, I think of Ammon definitely as a herpetologist, right? So Ammon did his PhD at UC Santa Cruz. He was working there with Barry Cinervo, and he's been working on Uda stansburiana biology, including the throat color polymorphism story and its role in sexual selection. And in fact, Ammon gave us a seminar here, an MVZ lunch seminar some years ago, so some of you may remember him from that seminar that he presented, although it predates many of the students, I would guess, because 2006 roughly it was, I think, when you gave that seminar. And then after he finished his PhD, he took a postdoc with Hans Elgren at Uppsala University in Sweden. And so he started working on this bird system that he's going to be telling us about today. And then since May, he's actually been back in the United States, and in fact, he's been sort of serving as an informal postdoc in the MVZ here. He's in the office next to mine. We've given him a home, and while he continues to work on this and other projects, and he's also, of course, he has grant proposals in the works to try to get a paying postdoc lined up, and building collaborations with Rasmus Nielsen and others. So for those of you who haven't actually had a chance to talk to him, he lives in that little office next door to my office on the other side of the MVZ. Check in with him. And then today he's going to tell us about his work on studying sexual selection and constructing species trees with genomic data in shorebirds.
Well, thanks for having me. It's great to be able to talk about my postdoctoral work. And so I'm going to get right into it. Except just to give you a little bit of my research background. This isn't going to be the focus of the talk, but just to give you an idea. So in general, I've been interested in questions dealing with speciation.
Sorry, Ammon, it's really hard to hear you in the back.
Okay.
Yeah.
I've been interested in questions about speciation, sexual selection, and how polymorphism is maintained and lost. Can you hear me now? Okay, good. And this is based on my thesis research, which was at UC Santa Cruz, and so I was looking at the polymorphic mating strategies in the side-blotched lizard, and basically what I did was I surveyed the geographic variation of the mating strategies. That's this map here, and then I used a phylogeny to understand how the species, how the different mating strategies evolved over time. And this is a whole talk onto it itself. If you're interested, please come and talk to me. I'm happy to talk about it. But it shows that my background really is a combination of both phylogenetics, studying sexual selection, among other topics. And that's what led me to my postdoctoral work. And so this talk is going to actually be two different topics. So the first one is going to be on sexual selection. So how does sexual selection affect genetic evolution? And for this, I'm going to go over some tests of a signature of sexual selection that sexual selection can leave in genomic levels of genetic diversity. And then I'm going to switch gears to another conceptual topic and talk about what's the best way to reconstruct a phylogeny with multi-locus data. And this is going to deal with some sampling strategies for looking at species trees. Now these are pretty conceptually disparate. I'm interested in both of them. Darwin was interested in both of them. He was thinking about sexual selection and Ruffs and phylogenies and things like that. But what connects them is the data set. The data set that I collected for the first topic, I could then apply to the second topic. And so hopefully it'll be connected enough. All right. So on to the first topic, thinking about the effects of selection, sexual selection on genomic genetic diversity. And the theory for this is actually pretty simple. So sexual selection is known to increase the variance in reproductive success. So for example, in some mating systems, a few males get many mates, and many males get no mates. That's a high variance reproductive system. And this polygynous mating system has higher variance in reproductive success than a more monogamous mating system. So in general, what variance in reproductive success does is it lowers the effective population size of the species, because if you have some males that aren't mating, their genetic variation isn't getting passed on. And this results in decreased levels of genetic variation. And then if males in particular have higher variance in reproductive success than females, as is often the case, then this lowers the effective population size of DNA that's predominantly transmitted by males. So in a mammalian system, Y chromosomes might have exceptionally low diversity because of this. And in birds, males have 2 Z chromosomes, whereas females just have 1. Polygyny on males may actually reduce Z diversity because of that. And overall, this creates a link between genetic diversity on the sex chromosomes and levels of sexual selection. So just to illustrate that in a little bit more detail, to give you an idea. So I've come up with some pictorial schematics of monogamy and polygyny. So you have males here and you have females here. And I'm going to show the case for an avian system since this talk is about birds. So in case you don't know, male birds have 2 Z chromosomes, whereas females have 1 Z and 1 W. In mammals, it would be 2 X's for females, and then an X and a Y in males. So in monogamy, what we can do is just sort of count up how many chromosomes are passed on when you have a monogamous mating system. And so each male has 2 autosomes and 2 Z's, and each female has 2 autosomes and 1 Z. And you can add all these up, and for these 3 individuals, you get 9 Z chromosomes to 12 autosomes, which gives you a ratio of Z to autosomes of 0.75. So the neutral monogamous expectation just based on copy number differences is you get 3/4, or 0.75, for a Z to autosomal ratio. Now this changes when you have polygyny. So if you have 1 male that gets 3 females and 2 males that don't, you can again count up how many chromosomes are being passed on. The male contribution is lower now, so it's 2 autosomes, 2 Z's, and overall you get 5 Z chromosomes to 8 autosomes for a ratio of 0.625. And so you can see that polygyny in this case reduces the Z to autosomal ratio. Now, in terms of thinking about effective population size and genetic diversity, there's some fairly simple equations for relating basically mating system to effective population size. And so just to give you some general predictions based on those equations, here are plotted the number of monopolized mates, meaning either males monopolizing females or females monopolizing males. And then this is looking at the effective population size of the Z chromosome relative to the autosome. So under monogamy, you only monopolize 1 mate, you have a ratio of 0.75. And then polygyny reduces this ratio in birds, and polyandry increases the ratio. Now, this is a ZW system for birds. In an XY system, such as mammals like us, the predictions are just reversed. So basically for polyandry you get reductions, and for polygyny you can actually get higher ratios than 0.75. But this shows in sort of a simple way how this metric is related to the mating system.
Doesn't this assume that there's only 1 autosomal chromosome? There's more than that.
There are more autosomes than that, but all the autosomes should behave basically equivalent to each other, so you can survey multiple different autosomes and get what's their level of genetic diversity and compare that to the Z chromosome. All right, so not surprisingly, people have looked to see, okay, they've collected data that can actually apply to this, and they've measured these ratios. And so this is actually genetic diversity patterns in humans, and this is how I got interested in this particular metric. Is as an undergrad, I've worked in the lab of Michael Hammer, and he has studied these questions for a very long period of time. And so in this study, he looked at genetic diversity in 6 different human populations, and looked at the X to autosomal diversity. And so you can see here, here's the prediction for sort of monogamy neutrality, 0.75. And across all the cultures, their average value is above 0.75. And in some cases, it's significantly above 0.75. And so what he argued from this is that this is evidence of polygyny at some point in human history, which isn't too surprising. But in some sense, the story isn't quite that simple. So these ratios can reflect sexual selection, but there can potentially be other things that can affect them. So the ratio of this, like Z to autosome or sex chromosome to autosomal diversity, is influenced by any force that basically works in a sex-biased manner, or that differentially affects the sex chromosomes and autosomes. So there's been an awful lot of theoretical modeling work on this, rather little data up to now, but the good thing for me is I could use these models to think about different forces. And so sex differences in mortality or sex bias migration actually have relatively minor effects on these ratios, whereas sexual selection actually has a relatively large effect. The one other force that seems to have a large effect on these ratios is changes in population size. So for instance, if you reduce your population size, this can give us a zeta autosomal ratio that's much lower, whereas a population growth can cause the ratios to actually go up towards 1. So from this, I'd argue, in some sense, it's hard to interpret any one particular point estimate within a species and say it's sexual selection. You have to control for other things. And this sort of thinking led me to my postdoc project. I was interested in saying, oh, maybe we can use these ratios in non-model systems and go in and say, hey, what's the mating system of these species based on just genetic data? But to do this, what we really need is a formal test of, does mating system influence these ratios in some sort of repeatable fashion? And so that's what I set out to do based on my interest. I did a phylogenetically controlled test of species that differed in mating system, and then looked to see what are these ratios to actually try and test, does it vary in a predictable fashion? So the basic data for this was I sequenced 13 Z chromosome and 15 autosomal introns in over 10 individuals per species, and that's just to survey sequence variation. And then I measured genetic diversity from the Waterston estimator of the number of segregating sites, which is known as theta. And in total, I obtained data for 6 species, 3 independent mating system comparisons. Now, it would have been great to do this in lizards, which I worked on before and I enjoy working on a lot, but the genomic resources for lizards probably aren't up to the task. They're coming along. But for birds, there was a lot more available. And the other appealing thing about birds was that there's a lot of diversity in mating system among closely related species, especially within shorebirds, and that's how I zeroed into shorebirds for doing my study. So this is looking at a phylogeny where I've mapped mating system onto the branches, and monogamy is here in blue, and then polygyny is in black, and polyandry is in green. And you don't really need to pay attention to the species, you can just see that there's been multiple independent evolutions of polygyny and polyandry within this group of birds, and there's been a lot of work on these shorebirds trying to figure out why that is. But what I was able to do was then get some independent comparisons of mating system based on this. And so I'll go over the players in this game. And so I have 3 different polygynous species and 3 different monogamous species. We have, on the polygynous camp, we have white rump sandpipers, pectoral sandpipers, and ruffs. And then over on the monogamous camp, we have leaf sandpipers, western sandpipers, and red-necked phalaropes. And the phalaropes, they're monogamous towards even polyandrous at times. Yes?
Is the species hybridized between themselves?
You mean between
Yeah.
pairs? They're closely related, but I don't think that closely related. I don't know of any hybridization, and Rory's shaking his head, so I'm going to go with no. But good question. All right, so that's the cast of characters, and in some of the later graphs, I just simplify things down. And so, say a western sandpiper is called by its first 3 letters of its name, so M-A-U. So here's what I found. So this is just looking at overall levels of genetic diversity. This is that measure of theta of genetic diversity, and here's the 6 different species. I've paired them, so this is an independent comparison from this one, and the polygynous species are underlined. And you can see, okay, autosomes always have more genetic diversity than Z chromosomes, that's to be expected. Phalaropes have the most genetic diversity, ruffs have the lowest. Keep that in mind, ruff diversity comes into play later on, and there's some sort of variance associated with these estimates. So that's the basic data, but what I was interested in were these ratios. And so what happens when I actually take a ratio of Z to autosomal diversity? Well, here are the ratios. And so again, remember that polygynous species, you expect ratios below 0.75. Monogamous, you expect 0.75. one of the things you may notice is they're all below 0.75. So if I only went into, say, the least sandpipers and said, hey, look at their ratio, I might say, hey, these are polygynous. But this is where I would argue that a really good way to do these sorts of comparisons is in some sort of paired comparative manner, because what you can also see is that in each of the pairs, the polygynous species has a lower ratio than the monogamous species. And so this is consistent with mating system actually altering these Z to autosomal ratios in some sort of predictable manner with mating system. So that was great. That went according to plan. And I could actually do a more formalized test of this. So I can look at independent paired comparisons here, and I can test for, does mating system affect these ratios? And sure enough, I get a significant p-value from that. So that was certainly nice. But I didn't really want to stop there, because if you think about these ratios, for the autosomal diversity, there's lots of variance, and for the Z chromosome diversity, there's lots of variance. And so you could ask for any of these individual ratios, are they actually statistically different from 0.75? So is this just noise, or is this actually varying in some sort of predictable manner? And so in order to test that, one of the things I had to do was I wanted to test, okay, are Z chromosomes statistically different from autosomes more than you'd expect by copy number? And so what I had to do was adjust for copy number differences. So Z is expected to be 3/4 the autosomes, so I multiplied the Z genetic diversity by 4/3. And so now the null expectation is that this should be equal. Hopefully you guys followed that. But that's what this graph is. So what I'm trying to test for is in each species, does Z diversity differ from autosomes? I've done a correction on the Z diversity so that under monogamy, they should be equal. And here's the 6 species, and then here's this measure of theta, and I've had to transform it to meet assumptions of normality. But when I did these tests, what was nice was that in all the monogamous species, so this one, and this one, and this one, Z and autosomal diversity were not significantly different. So even though they had slightly lower ratios than 0.75, those differences weren't statistically different. And so those species are perfectly consistent with the expectations of monogamy, like was predicted based on their mating system. All right, so what about the polygynous species? So 2 of them, this one and this one, so the pectoral sandpipers and the white rump these sandpipers, they showed significant differences in Z and autosomal diversity. So that's, again, showing that the predictions here are matching what the mating system predicted. And the one exception here are the Ruffs, which is sort of surprising. They're known to be quite polygynous, but when you do the statistical tests, you do not get a significant difference between the Z and the autosomes. But on the whole, like at least for 5 out of the 6 species, they went according to predictions. So I was also trying to think about, okay, again, there's lots of variability here. How else can I maybe control for things and look at the rough diversity or consider this? I was thinking about this in the phylogenetic framework in terms of independent tests. The one problem with that is that standard statistical models can actually, you can build much more complex model with sort of standard statistics that aren't phylogenetically controlled. And so for the next test, I turned towards standard statistics just to try and control for more factors other than phylogeny. And so what I tried to test was, okay, is there an effective mating system? And is there an effect of autosomes and Z diversity? And in particular, is there an interaction between mating system and chromosomes? So you'd expect there to be a difference between polygynous species in autosomes versus Z and monogamous species. But what I could do in this model is control for significant differences among individual loci in their genetic diversity, and also in differences among species in genetic diversity. And what I found was when I had all the species in the model, I didn't get a significant interaction. But again, thinking about, okay, the Ruffs seem to be doing something different. If I take those out and just look at those 5 species, sure enough, I get an interaction between chromosomes and mating systems. So the polygynous species have lower Z diversity than autosomes, relative to monogamous species. So overall, mating system is predicting something here, but what's going on with the Ruffs? So one of the things that's known is that when genetic diversity levels are low, estimates of theta can be inaccurate unless you sequence a really large region of DNA. And the basic reason for that is that short sequence fragments don't have enough informative sites to get a good measure. And the Ruffs had very low genetic diversity. So for instance, 2 of the loci had no segregating sites, and 7 had only a single segregating site. And so what I would argue is that the estimates that I got of genetic diversity may be relatively inaccurate because having few informative sites was common. One other thing to consider, as I mentioned, population size changes can affect these ratios. It'd be good to look at those. And one way to do that is to look at Tajima's D. And so negative values of this can indicate a population size expansion, whereas positive values can indicate a population size contraction. 5 of the species didn't show really prevalent deviations in Tajima's D, but one of them did. So the red-necked phalaropes, they're likely to have undergone a population size expansion because 12 of their 28 loci had significantly negative values of Tajima's D. But population expansions are supposed to lead to Z to autosomal ratios above 0.75. And if you recall, the ratio for the phalaropes was 0.69, below 0.75. And it's not statistically different from 0.75. So what I'd argue from this is that for all the species, really, population size changes probably haven't affected the estimates of these ratios too much. So just to sum up this first part of the talk, mating systems generally predicted the levels of Z to autosomal diversity, and this suggests that these ratios could actually be a tool for characterizing the mating system of the species, but low genetic diversity might be problematic for doing this. The other thing that what I think is interesting about these ratios is that what I would argue is the mating systems of these species must have persisted for a relatively long period of time. Is that we can go out and we can look at the mating system now, but how do we know that accurately reflects their mating system far in the past? Well, these Z and autosomal ratios actually reflect long-term changes in effective population size. So the mating system must have persisted for some period of time. And so I think that could be sort of an interesting use of these as a metric of persistent sexual selection. We don't have that many other than looking at, say, sexually dimorphic traits. All right, so I'm going to switch gears here. As I said, these are conceptually different but connected by the data set. But the other thing that I did with this data was to look at how best to reconstruct a phylogeny with multi-locus data. So prepare yourselves for another little mini talk. And just to catch you up on the general topic is, as you probably know, systematists want to resolve a species tree, which is the relationships among species in terms of ancestors and descendants. But typically this has been done by making gene trees. So this is what I did in my lizard work. I made a gene tree out of mitochondrial genes to look at the ancestor-descendant relationships of a particular gene. And from that, you hope to get some information about the species tree, but the problem is that sometimes gene trees and species trees match, and sometimes they do not. So there's various reasons for discordance between gene trees and species trees. Hybridization can mess things up. Gene duplication and extinction of different paralogs can cause a discordance. The main one I'm going to be considering here is lineage sorting. And this occurs when polymorphism in an ancestral population is basically retained across speciation events, and is stochastically lost in different lineages. And so this is problematic, especially in large populations, or when there's a short time interval between branching points and speciation events. And so just to illustrate this, I stole someone else's figure. And so this is looking at 3 different populations, A, B, and C. A and B are more closely related than C. We can look sort of a depiction back in time, and then you can sequence different individuals within each population. And so an ideal situation is you say sequence this individual from A, and you look, okay, this individual is next most related to individuals in B, and then it has a common ancestor back here that's related to C. And so you get this gene tree would correctly reflect the species tree, but it doesn't have to be that way. So if you sequence another individual in A, say this one, you can trace its ancestor back to this ancestral population, and it's more closely related to individuals in C than the common ancestor, which is related to individuals in B. And so this polymorphism in the ancestral population can actually cause discordance between gene trees and species trees. So how do you deal with this? Well, the basic way is you get data from more than one gene, but how do you deal with that data in some sort of way? And the way that people have started off doing this is just concatenate all the genes together, but there's been a number of studies that have shown that this can be inaccurate because it fails to recognize that the genes actually have separate histories from one another. You can reconstruct gene trees separately and use the most frequent topology, but sometimes the most frequent topology to evolve actually doesn't match the species tree, which is sort of bizarre. And just recently there's been a number of methods that are aimed at actually constructing a species tree that explicitly account for the stochastic history of gene trees. So one method, the one I'll be talking about, uses a multi-species coalescent model that considers gene trees in the framework of an encompassing species tree. So there was a recent review about species tree methods and the promise of them, and one of the things that was needed for further research was how should trade-offs among sampling longer sequences, more genes, and more individuals per species affect the design of multi-locus phylogenetic studies. And the argument was that sampling strategies for gene trees may be different from species trees. And there have been some simulation studies to address this, but when I started it, there was no empirical work on this. There's now one other empirical data set that's been applied to this, but I think there's a lot of room for exploring this topic. And the reason for this is, at least right now, there's been relatively few data sets that have enough genes or individuals to really address this question. But the Shorebird data that I collected did. The other thing I could ask is, okay, do different markers actually affect the resolution of the species tree? And so one thing I asked was, do sex-linked genes resolve the tree better than autosomes? And there's a reason to expect this, is that Z chromosome genes have a lower effective population size, which means a faster coalescence time, which actually means fewer problems with lineage sorting. And lastly, I was also interested in, does adding a mitochondrial gene to a set of genes actually significantly improve the resolution of the species tree? And mitochondria have a very low effective population size, they've got a high mutation rate, they could have a lot of power for helping us out. And there's some theory that suggests that adding a mitochondria can actually let you resolve a species tree with many fewer loci. All right, so here's what I did. So the first thing I needed to do was actually come up with, well, what's the species tree for these shorebirds? And so I used a program called StarBeast, and it co-estimates a species tree and all the gene trees in one analysis, a Bayesian chain Monte Carlo analysis. And then using data from 12 autosomes, 12 Z chromosome genes, and 1 mitochondrial gene, I constructed what my best estimate of the species tree was, and I call this the best tree in some later slides, just to throw as much data as I could at it and say, what's the best estimate of this tree? To get some idea of divergence times, I calibrated a molecular clock based on a published rate of evolution of the cytochrome oxidase mitochondrial gene. And then here's the tree. You've seen a version of this earlier. I'll go over it briefly. So all the sandpipers in the genus Calidris grouped together. We have white rum sandpipers, and we've got white rum sandpipers grouping with least sandpipers, and western sandpipers with petrel sandpipers, and then ruffs are next most closely related to them, and then phalaropes. But for the purposes of this study, what's important is that the support values for all the branches are quite high. So there was only one tree that was chosen in the analysis after all that. All the branches are well supported. This is hopefully a pretty good estimate of the species tree. So then what I wanted to do is test different sampling strategies. So I tested how varying the number of genes affected the resolution of the species tree. So I had sets of a single gene, 3 genes, 6 genes, and 12 genes, and what I did was I nested them inside each other to get independent sets in each level. So 3 single genes made one 3-gene set, and then 2 3-gene sets made one 6-gene set. You get the idea. And then I varied both the number of genes for the Z chromosome and the autosome. And for this, I tried to control for the amount of sequence based on those 2 types of genes. So the 12Z genes had about 8,400 characters. The autosomal genes had about 8,300. They had similar number of parsimony and formative characters. So they should have approximately equal power for resolving things. The next thing I did was I randomly removed one of the genes from the sets of 3, 6, and 12 genes and replaced it with a mitochondrial gene to look at the effects of that. And then in a separate test, I took the ones from this step and I took out the mitochondrial gene and put a Z-linked gene in the sets of autosomes to compare the same sets of autosomal genes but with a mitochondria versus a Z chromosome. And I chose a Z-linked gene for this. I chose this specific gene because it had lots of parsimony and formative sites. And the reason for this is that mitochondria have a high mutation rate. This mitochondrial gene had a lot of informative sites. And so I wanted to try and say, if you have an informative Z chromosome gene, how well does that do relative to mitochondria? All right, and then I measured performance of this by the number of trees in a 95% credible set of trees. And this is some measure of precision. So at the end of your analysis, you might get 10 trees out. And an analysis that gives you 2 trees out is sort of more precise in sort of what the topology is. And then I also looked at the percentage of the trees that match the best tree within that set. And that's some measure of accuracy. You might have relatively few trees in your set, but the most common one might actually not match the species tree. So this is what I'm going to show you in the next few graphs. And so this is looking at varying the number of genes, and this is the average number of trees in that confidence set, and then this is the average percentage of the best tree. And so you can see 2 things sort of right off the bat. As you sample more genes, your precision is improved, and the number of trees matching the best tree goes up. And so this shows that actually sampling relatively few genes, you can sort of converge on this best tree fairly easily. However, you can also see that the Z chromosomes do much better. So the Z is in blue here, and the autosomes are in black. And at all levels of sampling, the Z chromosomes are doing better than the autosomes. And I was actually surprised about how strong this was. And so, for instance, if you look at just 3 Z genes, they have about the same number of trees as 12 autosomes. And then these 3 Z genes has 70% of the best tree, whereas 12 autosomes, it's actually only 30%, and it didn't even match the species tree. And so what I took home from this is that these sex chromosome genes can actually converge on the species tree fairly quickly. So what happened when I added mitochondria? So that's these dashed lines. And so what happens is the autosomes go from here down to here, and the Z goes from here down to here. And you can see similar improvements on that graph as well. So actually adding a mitochondrial gene really helped resolution of the species tree, far more than any individual gene of the autosomes or the Z. And then the one last iteration of this was the autosomes plus the 1 Z gene. And you can see that adding a Z gene could help things, especially in terms of the percentage of the best tree. It didn't help things as much as when there was just a mitochondrial gene added, but it did pretty well. So there is some power to adding even just a single Z gene to your analysis. But one thing to notice is that even autosomes combined with mitochondria, which is here, don't do as well as the Z chromosome genes alone, and especially the Z chromosomes with mitochondria. So that's the best sampling strategy we found. And then, just to think about in general, why is there so much power here? One of the things that's known is that in terms of thinking about lineage sorting, it's most likely to be prevalent on time scales less than 5 NE generations. And sex chromosome genes will reach this it's not a sharp threshold, but it's a threshold. They'll reach 5 NE generations much sooner than autosomal genes. And another interesting thing is the utility of sex chromosomes in phylogenetics, just to warn you. It'll vary based on the mating system, so I'm trying to tie it back to the first part of the talk. So in species with female heterogameity, so birds, polygyny decreases the effective population size of the Z, and this actually increases the power for finding the species tree. In species with male heterogameity, like mammals like us, polygyny increases the effective population size of the X chromosome and actually decreases the power. So just to show you that, here I have the effective population size of the autosomes in millions, and then this axis is looking at how much sooner the Z chromosome reaches 5 NE generations than the autosomes. So how much sooner will it be past the point where lineage sorting matters a lot? And for at least the birds that I looked at, if maybe they had a million individuals for effective population size and monogamy, the Z chromosome will reach that threshold 1.25 million years sooner than the autosomes. So that could be useful, and polygyny is only going to help that, whereas polyandry will decrease the performance of that. Again, with mammals, that's going to be reversed. But it shows that the Z chromosomes, 1.25 million years is a pretty long period of time. So just to wrap up with one last analysis. So the previous analyses I showed you held the number of individuals constant. So I just had 6 individuals in the analyses, which was 12 haplotypes. And then in a separate analysis, I tested how varying the number of individuals affected the resolution of the species tree. So I constructed trees with just 4, 8, 12, and 16 haplotypes per species. And I did this for 4 different sets of genes. And the reason I chose these sets was it spanned a range of conditions based on the topologies that I had reconstructed when I had 12 haplotypes in the analyses. So for instance, the 6 Z genes, that matched the best estimate of the species tree with high confidence, whereas the 3 autosomal genes didn't match the best tree. So I was trying to look at this across a range of conditions. And here's what I found. So this is looking at the changes in number of haplotypes. This is the number of trees in the confidence set, and this is the percentage of the best tree. For Z genes, which tended to match the best tree, there was almost no changes as I changed the number of haplotypes. For autosomal genes, I got more changes. So for 3 autosomes, things look good on this graph. So okay, as you increase the number of haplotypes, you get more precision. But then when I looked at this graph, I was like, okay, when you increase the number of haplotypes, you have fewer trees matching the best tree. So there wasn't a clear-cut, okay, this is a good strategy. Sometimes it helps, sometimes it different, but in general, it didn't have large effects. So I tried to figure out, well, why is it having relatively minor effects here? In lots of the theoretical work, it seems to have a strong effect. And to do this, I had to think about the effective population size. So that can be measured from both the neutral genetic diversity rate and the mutation rate. And what I came up with was an estimate of about 1 million individuals for the different sandpiper species here. And some complicated math will show you that that's, 5 NE generations is about 5 million years. And if you look at the terminal branches on this phylogeny, it's greater than 5 NE generations, so it's about 8 or so. And so at that time period, you wouldn't expect much variation to be sorting within these species. And so when I looked at the individual gene trees, only 2 Z-linked genes and 3 autosomal genes still had intraspecific variation that was not reciprocally monophyletic, so it was still sorting. And so all the other ones that did have reciprocal monophyleticity, any of the variation that was among individuals didn't really help that much in estimating the species tree. And so I think that that's one reason why I was showing relatively small effects of the number of individuals here. All right, so just to wrap up that, so overall, I actually think it's important to think about these sampling issues when doing a phylogenetic study. I mean, traditionally it's been just sequencing costs. I mean, I'd argue sequencing is still expensive. It's dropping precipitously. but the computational costs with these analyses are really great, especially some of these like highly parameterized analyses that I was doing. And so the fewer genes that you can have in there, the faster your analyses will run. And so if you can get good loci to do this, it's worthwhile. And so what I would argue is people should think about using sex-linked loci in a phylogenetic analysis. And the reason for this is that you can sample multiple independent loci, each with a relatively low effective population size. And that can help in terms of lineage sorting. I also think that adding a mitochondrial gene to a set of genes can really help things based on this. And then sampling greater number of genes certainly helps. Sampling greater number of individuals, at least in my analyses, it didn't help that much. If you're dealing with much shallower phylogenies, it actually it's predicted to play much more of a role. So you can think about sampling more individuals there. So just to give you a final synthesis of everything, thinking about both parts of this talk, what I'd like you to think about is that the same data set can be used to test both for this genomic signature of sexual selection and to resolve a species tree. So if you're interested in phylogenetics, what I'd argue is maybe you should sample some sex chromosomes because that's good for resolving a species tree. But you can also use that same data to look at questions pertaining to sexual selection. And the only thing you need to do then is to sample multiple individuals. And the other thing is, in terms of getting this data, I did all this with Sanger sequencing, and it took a very long time to get the data. But fortunately, there seems to be some new sequence enrichment methods to use next generation sequencing. So in terms of getting this multi-locus data, I think it's going to be much, much more practical. And with that, I'd like to thank the people who made this project possible. My supervisor in Sweden was Hans Elgren, and he was provided a lot of the resources for this and a lot of help. I had lots of people that I talked to about this topic that were great. I couldn't do this project without tissue samples from museums and other researchers, and so that really made the project possible. And of course, funding is invaluable. And with that, I'd like to take any questions.